Conventionally, we are simply taught that a tRNA molecule is as the transport vehicle that delivers an amino acid to a ribosome for protein synthesis. But that teaching is very incomplete as it skips and combines certain steps in such way that it obfuscates important facts and further makes it seem as though ribosomes can join even unmodified naturally occurring amino acids into a protein. Now, what actually happens is that a loader protein, synthetase, burns ATP to remove the OH atoms, hydroxyl group, from the carboxyl group of an amino acid so that the relative carbon atom of that amino acid can be attached to the relevant carrier oxygen atom of a tRNA. Thereby, the loader is not only attaching the amino acid, as it also creates an unstable high-energy ester bond. Later, during amino acid delivery at the ribosome, that unstable bond, as created by the loader, proves to be exactly what the ribosome needs for its means of accommodating a peptide bond to form between the newly delivered amino acid and the growing polypeptide chain. At this point, you can think of a tRNA molecule with its tri-base anticodon as a barcode attached directly to an amino acid being as the content of a package. But if that unstable bond between tRNA and the loaded amino acid were to be exposed during transport through a wet or aqueous environment, the loaded amino acid would quickly break off from the tRNA due to the chemical nature of that unstable bond and the law of hydrolysis. So to ensure that the content stays attached to the barcode during travel, an appropriate type of package protection is required. Therefore, the EFTU protein. This protein physically wraps itself as a waterproof shield around the loaded end of the tRNA, and this protein stays wrapped around that loaded end even into the A site of the ribosome. And it is only once the ribosome has scanned the barcode and confirmed a match to the mRNA codon, then the EFTU protein becomes separated from the package. But note that as ribosomes are the only biological machines capable of protein synthesis, then how could these prerequisite proteins have already existed in a primordial pond or soup?